my great pleasure to introduce Dr. Judith Grisel. She is an internationally recognized behavioral neuroscientist with expertise in pharmacology, genetics, and her research focuses on the root causes of drug addiction. She did her bachelor's at Florida Atlantic University. She then went on to do a master's and PhD work at the University of Colorado. She did a postdoctoral fellowship at Oregon Health and Sciences University. When were you there? What year?
I was there 93 to 97.
I did a sabbatical there in 2001 and 2. It was awesome.
Oh, yeah.
I digress, sorry. And then, and she's now a full tenured professor at Bucknell University. She's won a number of awards, such as the Howard Hughes Distinguished Mentor, the C. Grayson Mary E. Rogers Faculty Fellow Award, and most recently, the Mind Science Foundation Distinguished Scientist Award. Her work has been supported by the National Institute of Alcohol Abuse and Alcoholism. She has published in top journals in her field, such as Addiction Biology, Alcohol, Behavioral Neuroscience. And recently, she has published a book, Never Enough, The Neuroscience and Experience of Addiction, which was on the New York Times bestseller list. And they say just wonderful. I mean, I went and started looking at all the reviews and everything, and they're just amazing reviews. I would encourage everybody to go out and take a look. Today, she's going to talk about Never Enough, how binge drinking informed a life and a career in neuroscience. And if we had an audience here, we could clap and say, welcome to Dr. Grisel. Thank you.
Thank you very much, Rick. It's a pleasure to be here. I would have loved, I think, the program, and especially to be away from campus at this time. It's our last couple of weeks of classes in person. And so this is, at any rate, a nice break. And I look forward to hopefully meeting some of you in person someday. So
awesome. I'm going to stop sharing my video so everybody's focused on you. Okay.
Okay. Well.
Okay. Just to let you
and
know,
they can
I'm here.
see the slides, right? The slides are up.
Yes.
Okay.
Yeah, they look great.
So I'm talking mostly about alcohol, but my interest, as Rick said, is to really understand what's different about the brains of people who go on to develop a problem before they ever start. And this is, you know, I'm sure you know, a tremendous problem. So 10,000 people every day die as a result of their excessive drug use, and a quarter of kids live in a house with addicted parent. And most of the people who develop a problem, like me, started using before they were 18. So my story, I've decided to start a little bit with that. And the book was, well, I'll get to that in a second, but I was a pretty typical, I would say, maybe better off than most child. I was 8 or 9 on the picture on the left, and I was strong-willed and well-resourced and full of ideas and ambitions. And I kind of on a whim took my first drink, which was a big one, about a half a gallon of wine when I was just around 13. And really it was this profound, almost transcendent experience that changed the trajectory of my whole life. So, you know, I'm here to talk about alcohol research. I became an alcohol researcher first hand, you know, at 13, you could say. And I drank as much as I could after that first time, and as often as I could, which was several times a week. And then naturally went on to other substances. So I more or less spent 10 years doing research on the street, I guess you could say. I was kicked out of 3 schools, the first one in 10th grade and then 2 colleges. So even though I graduated from FAU, it took 7 years. And I was also intermittently homeless. I contracted hepatitis C from sharing dirty needles. And by the time I was 23, actually, by the time I was 22, I hated myself. I had no self-respect, and nobody else really liked me very much either. I ended up in treatment, which this was in the 1980s, so I had no idea what treatment was. I thought maybe it was going to be like a spa to kind of give me a break from my crazy life, and it was not like a spa. It was more like a boot camp. It was a big wake-up call. I was told there that I had an illness that was killing me, and that if I wanted to live, I would have to stop using, which seemed like a terrible, terrible option. I mean, I really didn't want to live without drugs and alcohol, but after a few weeks with nothing in my system, I began to see that they were right. I wasn't going to be able to live if I didn't quit. So it sounded just, as every person like me gets to this place where you can't live with it and you can't live without it. And, it's sort of a terrible, but anyhow, I figured that there was a back door somewhere. So I thought that I would fix my problem by, if they say I have a disease, I'll just cure the disease and then I would be able to use, which is completely nuts of me. And I now realize it was nuts, but at the time it was the incentive that enabled me to try to change my whole life. Briefly, I was only going to do it. I thought it would take me 7 years to fix it, and then I would be able to use without self-destructing. So 7 years came and went. I finished college. I got into grad school. My life was about 1 billion times better, and I kind of have kept going. So I celebrated 34 years clean and sober last summer, and I'm grateful for all. Well, I was going to say all of it. Some of it's been kind of hard, but mostly I'm very grateful. And this is a summary of what I've learned about what was different about the brains or my brain from others who are not so destructive. I think it comes down to 3 main bins of influence. I've spent a lot of time studying genetic influences, and there are lots and lots of them. I'm curious especially about the genetic influences on personality. I don't know if maybe some of you saw this paper last week, I think, on height. So they were determining the genetic influences of height, and there's about 10,000 genes playing a role on influencing height. So addictions or substance use disorders are much more complicated than that, and there's probably at least 10,000 genes having an influence. But some of those involve things that I think happened for me. So high novelty seeking, high risk taking, very focused on getting rewards, and kind of insensitive to punishments. I can remember, after that first drink, I told you I was drinking quite a lot, and I was grounded about half my adolescence. My parents finally kicked me out when I turned 18, but before that I was grounded a lot. And my thought was, I'm just going to make up for it when I'm not grounded. I was not swayed by punishment, as I think many of us are not. There's a lot of environmental influences, including probably more than there are genetic influences, because that's also very complex. I think the access and opportunity and the early use played a big role for me, and development is kind of a recent interest. And I think the literature was probably ahead of me, but I'm now appreciating more and more how my very early use was probably one of the biggest determining factors. So I want to talk just about, we discuss what is addiction. I'm sure you're having good conversations about, well, everybody's virtual, so I don't know how many good conversations we're getting to have, but if we were in the hallways and at the tables, we'd be talking about it. And I have several definitions I use all the time, but this I have been betting lately. I think it most often begins just to experiment and quickly becomes a habit. That was the case for me, so that, you know, every Friday I could go to a friend's house. But what really turned the table, I guess, a little bit was using to cope, so that when I was frustrated or when I was sad or disappointed, alcohol especially quickly became a kind of medicine for me. And that led to compulsion and to withdrawal. And I think that the change from coping to compulsion is really one that is reflected in a shift from cortical to subcortical brain structures. And of course, with compulsion and regular use, we don't feel normal without the drug. And so that leads to a state of withdrawal when it's gone, and eventually despair, which is where I said I ended up hating myself. I think that these clearly are all due to changes in the brain that mediate tolerance and dependence and craving. And for that reason, you know, it's those are, I think, core attributes of addiction, along with denial. And obviously, it has to be deleterious to the individual and also to society. But I think most of my research is focused on trying to understand those. And especially, I'm going to share with you today some of the craving research. So you know, and probably some of you work on this, it's very possible to climb back up the steps, not maybe to the top one, but it took me not very long to go down, as I said, and probably I'm still coming back up. I still think about coping with drugs. It's sort of a natural state for me, maybe an unnatural state not to. But I don't think there's a lot of good evidence that you can go back to just casually using, so much to my disappointment. And I'm glad I didn't know that when I was first deciding to get clean. So just to kind of sum up, alcohol use is very common, and just a subset of people develop a problem. And I'm really interested in that neural vulnerability, not so much the consequences in tolerance and dependence, but in the neural vulnerability to having this slide into compulsive dependent use. And the rates bounce around a little bit. Lately, they're bouncing in the wrong direction, probably as a result of COVID-19 related stress and increases in depression and anxiety, which I can appreciate. But only this small group has a problem, and that's what my lab is interested in understanding. So today I'm going to talk mostly about research, and I have time for questions at the end. I decided to present research on one sort of genetic influence and how my understanding about the complexity of genetic influences to alcohol use disorders has really grown, and my appreciation for the complexity has grown. And the story starts with a old set of data. This was from Christina Giannolakis, and she and others in the mid-90s began looking at the relationship between the opioid peptide beta-endorphin and the risk for alcoholism. And I'm showing here a graph from one of her original monographs, but there's pretty decent body of literature on this relationship. But this one is particularly striking because on the left are people with no family history for alcoholism. And on the right, there are two or more alcoholics in 2 generations. So grandparents, parents, and kids. And these were in young people who presumably hadn't had alcohol before. And what you can see is that 2 things, I guess. One is that those with a positive family history have about half as much beta endorphin in their plasma, for whatever that's worth, with no alcohol on board. So they're deficient, perhaps to begin with. And even probably more striking, that deficiency is really ameliorated by alcohol administration. So there's not, there in general, there's thought to be a relationship between alcohol administration and the synthesis and release of endorphin, beta-endorphin, but that's seemingly more true, and it is more true in those who have a positive family history than those who don't. And this leads to this kind of self-medication hypothesis. And I'm not sure if it was the case for me, but I think my experience initially was so wonderful that probably, if I had to guess, I would think that I was one of those who had the strong response to alcohol. So beta-endorphin, as I said, is an endogenous opioid peptide, and it was discovered in the heyday of discoveries around the opioid peptides in the 1970s by Leon Smith. And it's a very potent agonist at the mu-opioid receptor, more potent than morphine. And it comes from the POMC or pro-opiomelanocortin gene, and it's the sort of last peptide in this large gene that also produces things like alpha-MSH and ACTH and the lipotropins. So it's here at the carboxy end of that protein. And the roles of beta-endorphin are, I'll show you the distribution in a minute, but a few important roles. It plays a role in energy balance and metabolism and stress homeostasis, which is what I'm going to be talking to you a lot about, and also in general rewarding properties of alcohol. But it is localized to just a few areas. So the number of beta-endorphin synthesizing neurons is pretty small in the brain. It comes from the arcuate nucleus in the hypothalamus primarily, and from there projects to other areas of the hypothalamus and the bed nucleus of the stria terminalis, which is a part of the extended amygdala having to do with stress and coordinating a stress response, and also the nucleus accumbens, which is obviously associated with all addictive drug reward, and the amygdala. Again, the amygdala and extended amygdala play a critical role in stress responding. The VTA, which is where this is also related to the rewarding effects of alcohol. And also, finally, the nucleus tractus solitarius down in the brainstem, having probably, well, playing a role in stress also. But in addition to the brain, beta-endorphin is in the pituitary, and there it helps regulate the HPA-axis. So the HPA-axis is obviously the hypothalamic pituitary adrenal axis, responsible for coordinating the neuroendocrine response to stress. And in this pathway, beta-endorphin inhibits CRH, and I'll come back to that. So it's thought that with stress, you activate beta-endorphin synthesizing neurons in the pituitary and also in the hypothalamus to inhibit or negatively feedback on stress response. So my question about the alcohol risk, so those people who have high alcohol risk and this sensitive endorphin response is whether or not the increased likelihood of drinking is due to beta-endorphin's role in either positive reinforcement, maybe acting in, especially in the connection between the VTA and the nucleus accumbens to modulate how much they really like alcohol, or negative reinforcement. And that is to say, how much does beta-endorphin play a role in contributing to the effects of alcohol to reduce things like stress and anxiety. So I thought we would start with that later thing. And the first set of data, I'll come back to both, but the first set of data has to do with alcohol's ability to reduce negative states. And there's an old literature in vitro here that shows that the more, I mentioned this earlier, but that the more, the higher the concentration of endorphin, the lower the secretion of CRH. So endorphin, as I said, inhibits CRH synthesis and release from those neurons that make that. And I study this in mice that are lacking the ability to produce beta endorphin. These were developed while I was at OHSU in Portland. And so I was kind of in the right place at the right time to study these mice. And like most knockout mice, or many, it's either a lethal knockout or there's not much going on. So we have found a few things about these mice. One is that when they're males, especially as they get older, they tend to get really heavy. And that relates to the effect I told you earlier, that endorphin plays a role in metabolism. But we've also been studying the effects of endorphin deficiency on the stress axis. So because beta endorphin is at the carboxy end of the POMC gene, these researchers, Malcolm Lowe and Marcella Rubinstein, introduced a premature stop codon so that there's normal cleavage and synthesis of the other POMC products, but no capacity to produce beta endorphin. And as I said, they look much the same. But one of our initial sets of studies involved whether or not low endorphin influenced the stress response. And again, the idea is that stress releases CRH and also beta endorphin, and that beta endorphin feeds back and inhibits CRH. And we found initially that, and then recently replicated, there was larger adrenal glands in animals who are lacking endorphin, suggesting that those adrenal glands are overactive. And we also found that corticosterone, the glucocorticoid analogous to cortisol in humans that comes from the adrenal glands as a sort of output to the HPA stress axis activation, is higher in beta endorphin deficient mice compared to beta endorphin wild type mice, which are C57 black 6, which is, by the way, the deficiency has been fully backcrossed onto those mice for the genetics people in the audience. So if they're lacking endorphin, they have bigger adrenal glands and higher levels of corticosterone in the blood. And that is especially true for females. So in fact, not so much in males. So there's a main effect of sex. We know this is not news because females generally have higher levels of stress and stress hormones, but especially so for the females lacking endorphin. And so our first studies were also looking not just at corticosterone levels in the blood by RIA or radioimmunoassay, but also by looking at gene expression, in particular in the amygdala. So we wanted to know, is Cort higher, is CRH also elevated? And I'm going to show you a lot of data about binge drinking, but I'm showing you here animals who were the controls for the binge drinking studies. And so these were C57 black 6 mice and beta endorphin knockout mice or beta endorphin deficient mice that were drinking water in a binge drinking model. And you can see that no difference in CRH levels in the amygdala, in the central nucleus, in the males, but in the females, there is a big difference. And that difference is also true in the extended amygdala. So here we looked at the BNST and the nucleus accumbens in sort of a large brain region. And in females, we found about twice as much CRH expression in beta-endorphin deficient females than in males. So in males, there was not a difference. So that is in the BNST and the extended amygdala. So this kind of brings me to the study of sex differences. And while I was looking at that, kind of going back through the literature, I came upon this quote from Alcoholics Anonymous, and I'm just gonna, this is from the Alcoholics Anonymous sort of textbook, sometimes called the Big Book. And in 1955 edition, at least, they said that they noted that female alcoholics have sort of a different profile from male alcoholics in that they have worse outcomes, and they have more quick outcomes towards alcoholism. So this is well known in the literature now, I think increasingly well known, even though men consume more than women. By the way, that's not true in rodents or in other animals. Women are more susceptible to alcohol-related health consequences. And this difference between the consumption patterns of human males and human females is narrowing. So it used to be that there was a bigger gap, and that gap is sort of slimming down, probably suggesting that either the causes for drinking are increasing if there's sex-dependent causes, which we'll talk about, then maybe those are more prevalent now, but also certainly cultural and social factors. So the fact that it's more acceptable for women to drink alone, for instance, or to buy alcohol, I think those consequences are also evident. So it's also the case that there's differences in the kinetics of alcohol, so it's absorbed differently and metabolized differently. And for that reason, the same amount of alcohol by body weight produces higher blood concentrations, which might explain why women develop problems more quickly. So, if they're both getting the same dose, women are going to have bigger problems than men. And this relates to the definition of binge drinking from NIAAA, which is in females 4 drinks in 2 hours and in males 5, because that gives about the equivalent blood levels, which is the blood level to get drunk. So binge drinking is really drinking to get drunk, which is the only way I ever drank, and still really can't see much point in drinking for some other reason. For me, it was a tool from the very beginning. And so binge drinking is a problem because it's increasing, and it's also a big risk factor for developing an alcohol use disorder, especially in young people. And I guess this is now the most common form of excessive drinking. And it seems obvious, I guess. People who drink excessively are drinking not for reasons other than getting drunk. It's been increasing more and more. Today, about 1 in 6 adults binge drink regularly, and not just 4 or 5 drinks, but usually more than that. And we think that the increasing prevalence, as I alluded to before, is partly due to changes in culture, and it's more evident in groups that are or have less advantages. So in minorities, in those who are disadvantaged economically, in women, in older adults, people who are maybe less powerful. And this is reflected, I think, in this interesting speculation that the increases are probably due to increases in stress and despair, and therefore more likelihood of using alcohol to cope. And again, using alcohol to cope as a tool is a good way to develop a problem. And I think as we're seeing, this was written before the last year of stress and despair or so. And so I think that incidence is increasing, and therefore the consequences in early deaths and family suffering are increasing. So binge drinking in particular can cause unintentional injuries, lots of things. Violence is associated with being drunk, increase in sexually transmitted disease, not that it's the fault of people drinking, but I think, less cortical control and more unintended pregnancies and poor outcomes for those pregnancies. Chronic diseases and cancers, as you probably all know, it's recently been clear, I guess 2019 in the Lancet, that even, one or two drinks a week has negative consequences for health and longevity. And really recently, like last month, I think there was a study in JAMA showing that just one loss of consciousness, so passing out incident, is associated with a twofold increase in developing dementia. So not good. It's a toxin, and certainly the neurotoxic effects are evident there, and it leads to alcohol use disorders. So it's a good predictor of developing a problem. This and because most people who develop a problem do binge drink, we could sort of calculate the economic costs, and people have determined that it costs about $2 every drink in the economic loss, in the reduced work productivity, increased health consequences, car accidents, legal fees, etc. So it's not trivial, which I probably don't have to say for you. So I study binge drinking in my mice in what's called the drinking in the dark paradigm. And I could kind of go on and on about this, but I won't in the interest of time. But I did spend many years, decades even, studying alcohol drinking in mice in a way that was probably not a great model. So they would rarely voluntarily drink enough to become intoxicated. And if they weren't intoxicated, what was I really studying? So I have a lot of papers looking at that kind of drinking. But in 2005, in John Crabb's lab, Justin Rhodes and John developed this drinking in the dark model. And it turns out that mice are very likely to drink enough to become intoxicated if the alcohol is provided only for a short time at a particular time of day. So the mice are nocturnal, they sleep when the lights are on, and then they wake up when the lights come off. And if you wait about 3 hours after the lights come off, give them a chance to groom and brush their teeth, then you open the bar. They hit it pretty hard. And they'll drink in this 2-hour period here enough to reach the 0.08 kind of legal limit. And so they really like it that way. And in a binge drinking model, what we do is have our mice in a cage with 2 bottles. For the first 7 days, those 2 bottles both contain water. Just to get used to the protocol, we come in and measure how much water they drink at the same time of day. And then in the first 3 days of a 4-day experiment, they get this decent concentration of alcohol in water. It's not all that tasty, for just 2 hours a day, as I described, 3 hours after they get active. And on the 4th day, we double the time. So the bar inexplicably for the mice stays open an extra 2 hours, and they drink quite a lot. They're used to it sort of being restricted for them. And so they hit it hard. And then when it doesn't get taken away, they go back to it. And some of them get, twice the legal limit. So they really do get intoxicated. And in my model, they always have access to water. So it's not that they're thirsty at this time. They are just going for alcohol. After the last binge, I euthanize them humanely and collect blood and brain and adrenal glands to measure the kinds of things I've been talking about. So the first thing that we noticed is that, this is supposed to say B6, sorry about that. In the type mice that have endorphin, the males and females drink about the same. maybe the females slightly more, but not significantly more in my hands in the Binge study. But if they're missing endorphin, then they drink much more if they're females than the wild type females, and a little less if they're males. So that here there's kind of an interaction in that beta endorphin doesn't do anything overall, but what it does depends on sex. And this is their average consumption. And whoops, let me see what that was. Well, oh, sorry, I did not mean to do that. Let me, it doesn't matter, I don't think. The same is true for that was their average consumption across each 2-hour time point, and then their binge intake is the same. So here is how much they took during the 4-hour period. And you can say this is 7 grams per kilogram in wild type mice. So it's a hefty amount of alcohol. They're, not, they couldn't drive. Let's put it that way. If they're completely missing endorphin, this is in a replication of the first study, the females drink more than the female wild types, and the males drink less than the male wild types. If they have half normal endorphin levels, so these are heterozygote mice, they're kind of a mixture, and they do have half as much endorphin as normal, we see sort of a dose-dependent effect of beta endorphins. So in females, the less endorphin they have, the more they binge drink. In males, the less endorphin they have, the less they binge drink. So our data was replicated actually right around the same time by a group at Rockefeller. And I'm just showing you this because I think it is a pretty robust interaction with sex. So this is from Mary Jean Creek's lab. And they found that in the same mice, there was a little difference in the wild types. They had lots of more animals, but that the females drank more and the males tended to drink less. So I think it's a pretty robust phenomenon. And I'm interested in why that is. And so we began to look at the HPA axis. And so what I'm showing you here is the levels of corticosterone first in the blood in males who were water drinking controls. So they had this same experiment done to them, but their second bottle was replaced with another water bottle. So all they ever drank was water. And you can see that corticosterone was a little higher in the knockout males, and that alcohol didn't really do anything to that. So maybe it raised it a little in the wild types, but there's not a big effect of alcohol on corticosterone in males. But in females, there is. So the blue line is something I sort of showed you this before. I showed you the Homecage cort levels, but now I'm showing you the cort levels in these water drinking animals. And they are elevated in beta endorphin deficient females. And that is reduced in beta endorphin deficient females who have access to 4 days of this alcohol for brief periods. So alcohol did really nothing to wild type males or wild type females or male knockouts, but it does decrease the output of the HPA axis in female beta endorphin deficient mice. So they have higher basal cort levels, and they seem to be able to normalize those by drinking. So we looked at the RNA again by qPCR from the brain punches of animals in this case in the amygdala. And what we found, I've shown you this, I think, already, so that the water-treated females have much higher levels of CRH that goes along with the higher levels of cort that I just showed you. And so what I'm going to show you next is what happens to that gene expression in animals who are able to drink, because alcohol reduced their circulating corticosterone. Does it also, and yes is the answer, it really dramatically decreases the, normalizes, in fact, the amount of CRH being produced. So this leads to our sort of interim hypothesis that a lack of beta endorphin results in a hyperanxious state. And I have other studies to kind of show that on the plus maze and the light dark box, low endorphin is associated with high anxiety. But that in females, the ability to drink alcohol voluntarily alleviates that state. And I'm going to follow up on that kind of in the rest of the talk in a few minutes by talking about things that also change as a result of the low endorphin. So as I said, the mice are fully back-crossed, and but we don't think that the effects on the stress axis are directly or solely due to the absence of beta endorphin, but instead kind of built into the system, the neural system that regulates stress and the stress response. But before I go to that, I want to follow up on the other hypothesis, which is to ask whether beta endorphin contributes to the pleasurable experience of alcohol. Is it mediating the positive reinforcing effects of alcohol? And I probably don't have to say this for you, but I'm going to focus here on the Mesolimbic pathway, which is part of why we're wired to use mind-altering chemicals. Every single human culture, and probably cultures pre-human, so Neanderthals, have used drugs to change the way they think and feel and behave. Every single culture that could, I should say. I guess the Inuit in the frozen tundra who couldn't grow anything weren't able to do so, but anybody who could did, and also other animals take mind-altering chemicals to change the way they feel. It's in across kingdoms, so birds and insects and cats take catnip. It's just very widespread. And this is because of this mesolimbic dopamine pathway that we all know and love, I hope. So the small group of neurons that project from the top of the brainstem in the VTA of the midbrain, the ventral tegmental area of the midbrain, to the nucleus accumbens of the limbic system, releasing dopamine, lead to a sense of thrill and pleasure that's kind of associated with news or something important. And so this system evolved through natural selection to promote eating and sex. And both eating when you're hungry and sex when you're not satiated is really pleasurable, and that's due to the ability of food and sexual activity to activate these neurons. Every single addictive drug, as far as most of us are concerned, is one that co-opts this pathway. But the drugs are more potent, obviously, because we can get them when we want them, and we can take them as much as we want. So even though we've been using drugs forever, we have not probably had addiction forever, because being able to get highly potent and compounds, whether it's alcohol or other drugs, kind of on demand, has really increased the likelihood of exploiting that pathway with drugs. So what about alcohol and beta endorphin and this pathway? And here we're measuring the effect of alcohol to cause beta endorphin release in rewarding, in the areas associated with reward. This is the nucleus accumbens, which is the limbic part of the mesolimbic pathway. And here's the VTA. And what you can see from other labs, including Foster Olive and the Genotype lab, is that using microdialysis, we can see that alcohol increases levels of endorphin in these key brain reward areas. Suggesting that alcohol should be, that beta endorphin, should play a role in the alcohol-mediated positive reinforcement, in the liking of the drug, not just getting rid of the stress response. And so I've studied that mostly, beside binge drinking, in classical conditioning model called conditioned place preference. So if you don't know about that, it's one of my favorites because it relies on classical conditioning, so it's fairly simple. In the typical model, you take a mouse from its home cage and you let it kind of run around this cage that has two distinct contexts. This one has these little wire floors and this one has this bar floor. And they run around for a day for about 10 minutes. And on the 2nd through the 9th day, so for the next 8 days, they get 4 injections of alcohol every other day, usually, and 4 injections of saline. And the alcohol might be delivered right before they get stuck on this side or this side, and the saline in the opposite. So what's happening is that you're associating the drug effects with one context or the other. And on the test day, you put them in the middle with the doors open and you just ask the mouse, go where you like to go. If you do this with cocaine or morphine, it's very reliable. So virtually every mouse or rat will go to the place that was associated with those drugs. With alcohol, not so reliable. In fact, I found it a total pain in the neck. I spent about, 6 or 7 years. I had a small grant from NIH to try to do the study in my mice, and I could not get reliable place preference. So I'll show you that, what I did get. So here is an example in females, and what we're measuring is the amount of time on the test day after conditioning that they spend on the alcohol paired side. And it should be more than 50% if they liked the drug, right? Because that means if they spent, let's say, 55% on the alcohol paired side, then they spent 45% on the saline paired side, then they liked it better. It was associated with some good feeling. So I did, I don't know how many studies with my undergraduate students, too many, summers, day and night. And I could not get conditioned place preference in even the C57 control mice. I went both to the 2 main people who are able to do this, Chris Cunningham and Larry Meadow, Chris at OHSU and Larry at MUSC. I worked with both of them, tried to copy theirs, and it would, sometimes show up like a teeny bit, but not reliably. So, and this is in females. It was even worse in males. What happened in my males was that, again, they didn't really like it if they were wild types. And if they were missing endorphin, they actually had a significant aversion to the alcohol paired side. So I kind of threw up my hands for a while, but it was bothering me that I had taken the money from NIH. And so years after the grant ended, I developed a single exposure conditioned place preference protocol, which if you do place preference in rodents, I would love to see you do this. A few people have been copying it, but it's a very simple protocol. The total amount of hands-on time, and this maybe if you're working with humans, you wouldn't be too sympathetic, but is an hour per mouse. There's no pre-exposure habituation. It's based on the notion that for people like me, the first time I got the drug, I loved it. So it's 1 exposure to alcohol and 1 to saline, either on day 1 or day 3. Nothing happens on day 2 and day 4. They stay in their home cage. On the 5th day, I test them. Here's my little box. So it's basically a very similar, it's just the floor is different. And now I did get conditioned place preference to alcohol, unfortunately, in all 4 groups, the same. This is the same dose that I used earlier. Now I decided to raise the dose, maybe thinking this is a study that we just did last week, and I have some good news about it. So in males, raising the dose, they didn't like it. So they were no longer different significantly from 50%. That was also true in wild type females, but not in the beta-endorphin deficient females. So again, these data suggest that if you're female and lacking endorphin, you like alcohol more. So we looked at the synthesis of dopamine in these key areas that we've been talking about, the VTA and the nucleus accumbens, in these mice after either water drinking or alcohol drinking. And we found, actually, was this in drinking? No, this is after an injection. And we upped the injection a little bit, and we looked to an hour and a half later. So basically in males, which are in red, nothing happened. So alcohol didn't do much, and there was no effect of genotype. In females, there was an interaction. And what's especially notable, I think, is that in the beta-endorphin deficient females, there is more tyrosine hydroxylase activity after alcohol than before in the accumbens and in the VTA. So I have a lot more data, but I realize I'm kind of out of time. So my data was on GABA, and I think I don't have time to really talk about it, do I?
You could definitely, if you want, we could maybe, if you wanted to present for the next 5 minutes or so, that would still leave us 15 minutes for questions.
So yeah, I'll do 5 minutes quick, and then maybe the questions will be good because it's getting maybe more interesting for some of you. So I think I told you that the beta-endorphin deficient males drank less, and I haven't really addressed that. One reason we think they drink less is because they are very sensitive to the sedative effects of alcohol. This is a whopping dose of alcohol. We measure how long do they pass out for, which is called the loss of writing reflex. No difference in genotypes in the wild types, but the female knockouts wake up quickly and the male knockouts wake up slowly. And this led us to look at another protein. This is a subunit of the GABA-A receptor, which is known to play a critical role in the anxiolytic effects of alcohol, the effects of alcohol that reduce anxiety. And it's also been associated with alcohol use disorders in humans. So we looked at this alpha-2 subunit to test the hypothesis that maybe there were sex differences in the expression of this subunit such that females went toward anxiety reduction and males went toward sedation. So they went through the drinking in the dark protocol, and then we looked by qPCR at this subunit expression in the nucleus accumbens and the VTA, but while we were there, we also looked at some stress-related areas, the extended amygdala and the central nucleus of the amygdala. And in males that drink water, the beta-endorphin deficiency has resulted in a decrease in GABA-A2 expression, and the same in females, kind of a really dramatic decrease in females. But if they drink alcohol, so these are the ones that self-administered alcohol, the males can kind of bring it up to normal nearly. And the females, I circled this because I had to change the axis, not only get up to normal, but way past it. So it does look like by taking out endorphin, there is a sexually dimorphic response in dopamine pathways and in CRH and in GABA alpha-2 subunit expression at least. So missing this single gene is enough to change kind of the whole constellation of the brain in how it responds to stress and how it responds to the pleasurable effects of alcohol, suggesting that females are more sensitive to negative reinforcing effects and males more sensitive to locomotor sedating effects. But either way, missing endorphin modulates CRH and the stress axis, as well as the alpha-2 subunit of the GABA-A receptor in a sex-dependent fashion. And I think this suggests that we need to maybe look at how prevention and treatment efforts in humans might need to also be sexually dimorphic or reflect the fact that, at least in animal models, the reasons for drinking may diverge. And this is just a few points about studying sex differences. And, I am guilty in the fact that all through graduate school and most of my postdoc, I studied only males, male rodents, rats and mice. Even though right in the middle of all that, Robert Plomin and anyway, McLaren Plomin suggested that there were likely to be sex differences in the genetic influences based on sex. So they did early studies that showed that. But despite it, you've probably, hopefully seen this great review paper from about 10 years ago, where Beery and Zucker pointed out we're really not studying females enough. So the AA book knew it, the geneticists knew it, people who had their eyes open recognized it, but we really have not been studying males and females enough and comparing males to females. And this is just my attempt at looking how that's changed. So the view of sex differences is really increasing. I'm sure if you broke this 5 years out, you would see that it went up every single year. I think it's a really important area for research and also for the clinic. And I'll just end by pointing out that all the research that I've showed you has been done with the help of undergraduate students. I'm at a primarily undergraduate institution, and I have had 2 grad students now. One is still with me. The other is Todd Nenwig, who is now at MUSC in the PhD program, and Madison Waldron, who's looking for a PhD after next May when she graduates. So I also want to thank NIAAA and my collaborators at Susquehanna University, including especially Dr. Erin Reinhardt. And thank you for your attention. That's it.
Thank you so much. I'm going to do a clap from everyone. I appreciate it.
Listening to.
So we can definitely we have about 15 minutes. So if folks want to put questions into the chat box, we can definitely ask those and make sure we get those answered. I will probably ask Rick for some of your help, depending on exactly how technical any questions are. I definitely struggle through those, not my area of expertise.
Should I unshare? Is it okay?
Sure, if you'd like to. Yeah, absolutely.
Just
then we can.
So if maybe I could see, I mean, I don't know if there's too many people, but if anybody wants to show up, but you can't see me.
Yeah, I think we just lost your video.
I'll fix that.
Yeah. There we go. Perfect. Sounds great. Awesome. So I'll let maybe Rick, you start with the question, and anyone else who has, not to put you on the spot, but I'm sure you have more than one. And then anyone else who has questions can throw those in the chat and we'll get to those time permitting.
I do have a history, don't I, Devin? I have questions. That was wonderful first. Thank you, Judy, very much. That was great. So I'm going to start at the end. I love that single experience place conditioning experiment you showed, and it got me thinking about smoking and nicotine in that sort of the first experience with the cigarette and the subjective effects that humans retrospectively report, right? Because you're asking them as adults usually, or well after they've had their first cigarette. That predicts whether they're going to go on to be smokers or not. And it doesn't matter whether it's positive or negative, like I puked my guts out and blah, 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 or I felt euphoric and lightheaded. And I was wondering, is there anything like that in the alcohol?
There absolutely is. So that's a good point. I probably should have said that it's a very predictive in the clinic. And it began with Mark Schuckett at UCSD, who showed in the early 80s that the initial subjective response to alcohol was one of the main factors that predicted. So people who were stimulated and not so much sedated, so it does matter a little bit, were much more likely to go on. And Laura Ray in Northern California now has gone on to really expand this. So she's looked at the genetics of it, but this initial subjective response, I think, is, my hope is that I'm able to, I want to go back and do some QTL analyses now that I have a good phenotype, I think, or I think the phenotype is good. I have done the single exposure in alcohol and in opiates and in benzos, but I have not in nicotine. I bet it would work very well and it's really a high throughput screen because 20 minutes with alcohol I guess nicotine absorption would be just as quick if you're going, would you give them IP?
It's very fast. Yeah, it's sub-Q.
So I inject, I take them out of their home cage, I weigh them, I inject them, I stick them in there for 20 minutes, I pull them out, and the 20 minutes is based on sort of getting the peak levels, the peak brain levels when it's most euphoric. And then on the test day, I usually give them 30 minutes just to show it, but automatically recorded. And so literally each mouse is, an hour and 10 minutes. And we made the cages ourselves. I could give you the recipe from Home Depot, and it really works well. And another point about that is, I believe that the old model works if you're not so clumsy. But I work with undergrads, so I have all these people in my lab who, are just learning, and it doesn't matter. So this is really reliable for me. They, spend about 60% of the time on the alcohol paired side, and 40% on the saline paired side, and it's virtually every mouse. My ends are like usually 8 to 10, so it's very consistent. By the way, I've seen genetic differences with opiates and with benzos, but not so much with alcohol yet.
Interesting. You mentioned Chris Cunningham. I went there for sabbatical to work with him to learn the alcohol field. So it's very cool. You have a couple questions, I think, coming in the chat. You want to read them?
Yeah, thanks so much, Rick. So it looks like we have a question from Ken Wakabayashi. He said, excellent talk. Thank you. I was hoping that you could give some of your perspective on whether your findings can have translational relevance in terms of highlighting a novel or underappreciated therapeutic strategy in people.
Oh boy. Well, this is the important question, obviously. I can kind of get lost in, the details of this. I think the one big point is that it seems that if women are using more to cope with stress, which may be related to heritable levels of endorphin, then strategies focused on mitigating that stress or dealing with that stress in other ways would be more effective in them than maybe in men. So I think treatments and prevention should be somewhat sexually dimorphic. Now, I'm also in the middle with my grad student now, Madison, of looking at the effects on age. So age effects. So I don't know if young mice and humans sort of start out more similarly. I do know, and this is so non-scientific, but my anecdotal response was that this first drink was one of the best things that ever happened to me. I thought, it was really so great. And so can we, your question that I'm kind of skating around, a novel or underappreciated therapeutic strategy. Yeah, I mean, we're all looking for that. I'm a little skeptical, I guess. I feel like translation hasn't happened as quickly and as efficiently and as clearly as any of us would have hoped 30 years ago or 20 years ago or 10 years ago. And I'm still at the stages of trying to understand the system. And I feel like eventually, by better understanding, we'll have better strategies. But I was telling this to somebody, not a scientist, the other day in a talk somewhere. They said, well, could you use another drug then to fix the beta-endorphin levels? And I said, yeah, morphine. I don't think that's the kind of translation we're looking for. So anyway.
Thanks so much. It looks like Tierney Lorenz has another question, also mentioned incredibly interesting. Thank you for your personal perspective. She asks, your findings on the sex differences of HPA involvement are really compelling. Given your findings on anxiety relief versus sedation effects, I'm curious if any of this work has extended to looking at the ways that alcohol influences crosstalk between SNS and HPA stress responses. I ask because of the work showing even bigger sex differences in SNS responses than HPA responses, at least in humans.
And SNS, I'm sorry, Tierney, what is that I should know? Somatic nervous system? No. Sympathetic? No.
It's a great question.
Sympathetic.
Okay, good. I'm
Thanks,
sorry.
Tierney.
Yeah, I should probably know. It's just, I just gave a talk, so I can't think. So I think it's connected, right? So the core levels would be, it's it's easier to measure than ACTH, and I haven't looked at adrenaline or um, I don't. I guess I'd be curious about what you think might be a good uh, proxy for activation of the sympathetic nervous system. But I feel like it has a heart rate. Oh gosh, that's so simple. I love it. No, I have not looked, but I think we should do that. You and I, uh, or you, or me. But that's a great idea. It would be really simple. So look at heart rate in um, male and female knockout wild type who are just taken out of their cage or in their cage, and those that have alcohol on board. Yeah, let's do that. Okay. Well, we'll should talk later, me. But I think it's a great idea. And I love the thing, I have this problem of sometimes assuming that the rest of the body is just there to hold the head up, really to hold the brain up. But that's why I like that adrenal thing. A biologist friend predicted that. And I would have never thought to look at the adrenal glands. But now we're going to look at the heart rate. And I know there are good ways to measure that in rodents. So I'm excited. Telemetry. Good. Thanks.
Thanks, Tierney. Great question.
I was going to piggyback on that because I was thinking about a couple things, that hyper anxious state that you were referring to as your sort of hypothesis for the lack of the beta endorphins. And I was wondering two things. One was, have you tried to simulate that hyper anxious state in an animal model and then study drinking so that you're in control of the state? And then
yes.
I'll ask the second part next, because I hate when someone asks two parts and I don't remember the second part.
Oh, you were going to wait. Oh, that's so nice. That's a great thing. So I have tried and tried and tried. And in fact, this began when John Krabs sent me to the Jackson Labs, I think, or maybe NIH, to study the relationship between stress and alcohol to try to understand it. And I sat in a room I was a postdoc with brilliant scientists whose work I had all read. And they argued for 3 days, about the relationship between stress and alcohol. And we still don't really understand it now. At the time, I was like, what? I'm spending my career on this and nobody knows what it is we're studying. But we know that anxiety disorders in humans and depression, sort of the other side of that coin, are comorbid with alcohol use disorders. So stress dysfunction is predictive of excessive drinking. And we also know that excessive drinking leads to stress dysregulation. So they're clearly involved, but in most of the models that I've tried, which are many, the stress decreases drinking. And this is true for other people too. Howard Becker now has a model, but I've tried, wolf pee and loud noise, and I'm trying to even remember, lots of things. And it's not so terrific in animals. So I do think that the DID is a little stressful. And I say that because every time I go on a diet, I gain weight. And so this thing for the mice, that they can only get it for 2 hours a day, I mean, it seems to really tick them off. They as soon as it comes, they go, and then when you take it away, they kind of look like they're glaring at you. So I think that the restricted access, Kathy Grant in Oregon also studies this not being able to control your own administration is a good stressor. So I don't know if I have tried, for instance, restraint stress before the condition place preference, and it didn't do anything. And I may have not tried it with the right parameters, but I haven't found that. Now, one thing I did find that is a little bit exciting is I took Swiss Webster mice, which are like the Smiths of the mouse world. So just outbred populations, your genes are all over the place. And I categorized them as I put them on the plus maze and the light dark box about a week apart. And then I did a median split on their sort of basal anxiety levels. And then I did the condition place preference protocol, and the ones who were high anxiety had stronger CPP significantly. And I have to replicate that when I can get more students back in the lab and we don't have to socially distance, but it may be that shows up. So the second part of your question?
Well, so you answered the second part. So you weaved it all in there because I was going to ask about the human work related to it. So you tied that in nicely. One thing I've thought about is is because nicotine and smoking also are related to stress and the HPA-axis. And I've been wondering, we've never done it, never done any of the work, but thought about what would happen if we implanted a cortisol pellet so you have sort of constantly high basal level cortisol. Would that change their perception and the effects of nicotine over the long term? But not done that.
Yeah, it's interesting because alcohol stimulates cort, and then but is used as a sedative and to relax. So it's also a stressor. And nicotine is the same. Yeah, because the acute. Yeah, so it's really interesting. I'm an ex-smoker too, so I can appreciate the anxiolytic effects. And still, 25 years later, I have a stressful day and I think, geez, I'd like a cigarette. Luckily, my husband hates them, but yeah, I think that stress is so, it probably influences everything. And in that way, it's not going to explain much. It's just that we have to, I think, use our models in a way that's sensitive to variation in the stress response. So I'm afraid it can't be more helpful. I do think probably people know that females are 2 to 3 times more prone to all stress-related illnesses. I showed you high levels of cort. That's true in mice, that's true in rats, that's true in humans, that's true in monkeys. So females are just more stressed. And Deborah Bangasser at Temple has some really cool data showing that on the CRH-containing neurons, when male neurons in vitro get cort, the CRH receptors internalize as a way to sort of dampen their stress response, and females proliferate. So it's almost as if evolution has selected for this, one insensitive or dampening sensitivity and the other increasing it. But this reason is, or this neurobiology is thought to help explain why women are more prone to anxiety and depression and autoimmune disorders and all stress-related disorders. And whether that has to do with beta-endorphin overall, I would really love to know, because we're not all equally stressed out. Yeah.
Indeed. Well, thank you so much for your talk. I know we're
You're
at
welcome.
time, and I want to be respectful of your time and all of our attendees as well. So it was truly a pleasure, and I do very much, hopefully we'll be able to have you join us here in Nebraska in person, and we can have some of these conversations and follow up as well. So from all of our attendees, virtual clap.
Yes.
And
Thank
thank you so much. Thank you everyone for attending, and I hope you'll join us for the poster session coming up next as well. So thank you again.
Bye.